me start recording before I forget. Before we begin, are there any questions? Any questions about issues? Again, as I said, I'll try and get your grades to you as soon as possible. Uh, I think I have some of them in. Let me just check, but not all, obviously. Um, Yeah, other than one, and then for homework too. So basically, what I'm doing is I'm going down down my email from latest to earliest, and as I find the homeworks, I will. So I appreciate everybody's efforts. It was one of the nice parts about doing the homeworks and keeping them open-ended are there is not again and just again we go back to this idea of when you do research is you do not this is not a 2 plus 2 equals 4 scenario, right, where you have answers to questions, right? This is a research scenario, and I think that almost every one of you had a different approach, used different software, had some actually a kind of a different philosophical a different philosophical approach to how you even began this process. So I think that actually that is very good. There are no fixed answers, and so I think that that was actually, in my opinion, excellent. That you each came at it from a completely different standpoint. Okay. I The other thing is I actually went to check on the David, that resource, and it's still not allowing me to go into it for security reasons. So I don't know. I guess we'll have to let it go. Okay. All right. If there aren't any questions, let us go to, again, as I said, I'll keep the chat open so I can see what's going on, and so we'll go from there. All right. So now, when you begin, we got an introduction to kind of the genomics side of bioinformatics that you can learn a lot. And if you went to the different, the genome browsers, the fundamental difference between Sanger sequencing and the Maxim Gilbert sequencing, and some additional things. Certainly you learned how to use the strings DB, which might have seemed a little out of place with the kind of topic, but I thought this would be a good time to introduce it. I was actually, I think 4 years ago, I was in Malta just around the time uh, the effects of uh, of COVID were beginning to be felt in Italy, and Malta is about a 3-hour flight or so from or down down south from Sicily in Italy. And so of course I flew in via London, but I remember giving a talk for a completely unrelated thing, and I just mentioned um, the strings DB that we introduced last class, and I got a very interesting response. That is, somebody said, "Hey, I've never heard of it. Sounds very interesting. Can you tell us what the link is?" So I just said, go to strings DB, and you should be able to find. And you can see what a nice little network of information you can create, and the lines that connect the nodes, each of which represents a protein. It's a protein sequence, kind of a network system, and in which you can actually identify how those connections were made, right? Either experimentally, or something that was archived in a database by humans, or something that a artificial intelligence software extracted it from the text of the literature and things like that. Okay, so now we're going to go into something a little more topical. And we always talked about how bioinformatics, we use the example of the six blind men who were trying to figure out what an elephant looked like. And so uh, when Lincolnstein, and this is, you can refer back to your first, where he said bioinformatics was dead, and because they thought they did everything they needed to do at a sequence level, until they realized that it was a completely different ball game when you scaled up to to things at a genomic level. Okay, and then also what happens was you got a whole bunch of instrumentation that began to pop up, and we looked at microarrays, which was the first first chance at doing kind of a genome level, a tissue level, an organ level assessment of different genes, different conditions, different comparisons. And so the idea was then, well, we need to get better, right? And so right now, if you look in bioinformatics in terms of its applicability, you'll always look at something called as next generation sequencing. Okay, so next generation sequencing, let's see why. So again, those of you who are interested in next generation sequencing at a kind of a more in-depth level, there is a training course at the European Bioinformatics Institute, the EBI, and this link should take you to a whole bunch of videos that depending upon your personal interests, you may or may not need. But I thought I'd throw that in as a placeholder so you can go and learn a little bit more within the context of this class, within the scope of this class. You could use I wanted to introduce you to next generation sequencing and then give you a little bit of an example of two different kinds of more popular next generation sequencing. Though that changes, different people have different examples, but this is just to give you kind of an idea of next generation sequencing. Okay, so we look at some real time data, and you'll get an idea, and the homework will also probably be based on something like that. Now, again, for this is just a bit of a teaser. If anybody wants to learn more in-depth next generation sequencing, we have a class that is taught in spring. Okay, and again, I don't have the class number. Actually, I could. Let me just find out about. And so you can look at the web page here, and you'll see that we have something called as BTEC 5312. It's it's called gene expression analysis, where Dr. Sunita Sukumaran, the faculty in the genomics in our genomics core, will take you through let's see here. So for example, you will see differential gene expression, RNA-seq technology using next generation sequencing method, and then you'll perform NGS, you'll quantitative real-time PCR. You'll also get a chance to see, go through the entire sample prep, and you'll get to see, in addition to sample prep, you'll also get to see uh, you'll be able to get to use the what is called the MySeq instrument, and around you will be a whole bunch of other instrumentations like long read sequencing that we have acquired from PacBio, the workhorse, which is Illumina, the Illumina instrumentation that we use for regular reads, and then we've got two other instrumentation that we've recently acquired. One is from PacBio, the Onso, and another piece of instrumentation that we've acquired. Okay, so there is a whole bunch of things. Granted, these are 600 to 1.2 million dollar instances of, but you will definitely get a chance to basically go through a sample prep process and load the lanes on a MySeq. Okay, Dr. Mohammed Fokar, who is a genomics core manager, also teaches a summer class. I don't know if anybody's taking it right now, and we'll just go and do a quick search on BTEC transcriptomics. Okay, so yeah, that's called BTEC 5001. I think it's very likely that this number will change. It was because it was basically a special topics class, but principles and application, and this is transcriptomics. So these are the two courses that are taught, and then so this designation, the BTEC designation, might change, and then we have another course where you actually get where you actually can work with mice. And then again, you can see that this 5001, these are now dedicated courses, but as we go on, you can look at animal genomics at Texas Tech University. which is taught. All these courses are taught in spring. I think transcriptomics is taught in summer. I don't know whether Dr. Fokar is teaching it this semester, but these are all the courses that come arise from our center, where you'll get a chance to learn a lot of like literally hands-on use of instrumentation. We take you through the entire process of sample prep and things like that. The So at least I transcriptomics, gene expression analysis, and animal genomics. And those of you who want to the gene expression analysis also has a bioinformatics component to it. In addition, then we also have, not us, but in the Department of Biology, we have a course, Biol Genomics is taught by David Ray. So we can see that's taught in fall, because you guys are taking a summer class, so you can actually see that you can ask him about this class where he teaches a genomics class, which also has a bioinformatics component to it. And then you have Caleb Phillips, who teaches a class whose expertise is in metagenomics. His expertise is in metagenomics, and but he's a pure bioinformatician. So he will also teach a class in fall. So again, those of you who are inclined to be interested in all the training that we have to offer, at least I've named you 5 classes. And somewhere along the line, if you forgot what I just said because it's not part of the slides or the slide deck, feel free to email me and I'll be happy to point you in the right direction. Okay, at least this is what I know. I'm quite sure there are other courses taught in other places at the center. Okay, at the center and departments, and I haven't even touched on a lot of genome level work, especially related to the clinical side, is being done at the TTUHSC. Okay, and so that is another place that you can recourse to. Okay, so again, a lot of training that you can lot of training that you can acquire by yourself, a lot of training that you can acquire online, a lot of training that you can acquire by taking courses at our center. Obviously the advantages of, and I'm not necessarily pushing our courses, I'm just saying that if you want real instrumentation, you can take our courses. I'm not familiar with what experimental infrastructure is available for the biology courses. At least metagenomics will not have it, but Caleb does a great job in teaching the bioinformatics of it. And so there are many opportunities for you to study and get hands-on experience. Okay, it's kind of an interesting anecdote. We had a student once who actually went to work for one of the big pharmaceutical companies, Boehringer Ingelheim in Connecticut, and he emailed me and told me that their department had just acquired a MySeq. And MySeq is such an old instrument for us. We don't even use it for actual, other than for teaching purposes. We don't actually even use it for any kind of our research services or any kind of research project. We very often we just the samples, because ours is a service research center, people send us their samples, and we often make sure that that we can do a quality control. So it's just used for quality control. It's a small machine when you compare in terms of the number of reads, etc., that are processed, the number of samples that can be analyzed, and so uh, that is a very that is just kind of like a starting machine, just to make sure that the sample is fine. Obviously we have kind of right now acquired state of the art instrumentation, and so not to boast, not to brag, these but again, the infrastructure for your training and education is there. Now some of you who have in terms of using of the more modern instrumentation, we also, and again, this seems like a kind of an unnecessary teaser, but I'm going to say this anyway, is that if your mentors, etc., come to us for research services, then as part of the research services, that you pay for, not necessarily salaries and things like that, but you pay for our basically things that cost, right? Reagents and things like that. That's what you pay for. And so sometimes if your mentor says, "Hey, my student would like to be trained," then as part of that service, we also include training. So that research project, that your mentor's research project, can be done by the student with the right training and all of that, and we can just use it as a and this is something that you put in your resume as, oh, I've been trained on this instrumentation, I know how to use this instrumentation. It's easy to slide into a job with that kind of training. Okay. All right, let's keep going. So again, we go into what is sequencing. The question, the answer to that question still remains the same. It is the exact same thing which happened in the second slide of this class, and that is, you have a protein, you have a DNA. In proteins you have your standard 20 amino acids, but the combinatorial challenges are, because we don't know what the order is, and when you add size, remember I used the factorial in statistics. 2 factorial is 2. Two things can be, A and B can be written in 2 different ways. But the moment you have A, B, C, it can be written in 6 different ways. And whenever you have A, B, C, D, it can be written in 24 different ways. And when you have A, B, C, D, E, it can be written in 120 different ways. And when you have A, B, C, D, E, F, it can be written in 720 different ways. And so and so. So basically, sequencing is a very challenging concept, and the reason it is challenging is because that because of the combinatorial challenges. More importantly, when somebody comes to you, they say, "Here is my sequence. It's a small file consisting of a few microliters of a colorless solution." Now you have to start with that colorless solution, and you have to say exactly what order the DNA or the protein sequences are, and which is why somebody like a Sanger and then somebody like Maxim and Gilbert share the Nobel Prize, because they figured out a way of how to do it. Methodology is completely different. One is a destructive methodology, which is mostly derived from organic chemistry. That's Maxim Gilbert. And then you have a Sanger sequence, which has more biochemistry origin, and you can actually build a complementary sequence to your test, to your target sequence, without really destroying that sequence, right? And so you can go back and look at your notes of dideoxynucleotides, nucleotide triphosphates, and chain propagation and chain termination and things like that. Okay. So again, you can look at this video, and again, I just kept this as placeholders. So Sanger sequence was used to determine of a nucleotide sequence, one sequence at a time. Okay, and you can go and look at this video. It's a really nice graphical representation. In next generation sequencing, you have pretty much several hundred million next generation sequencing experiments happen at the same time. Okay, therefore it is also called massively parallel sequencing, which means they happen parallelly, and then you can then put together. So each of these sequences are broken into smaller pieces. Each of them are sequenced separately, and then they are put together like a jigsaw puzzle to form a long sequence. Now, that your traditional sequence will rely on especially when we talk about next generation sequencing, we are looking at genome level instances of information. This is why we have to this is why we have to make sure that you have a genome. Then we will also look at something called as a de novo sequencing. A de novo sequencing is where you have your sequences, but it does not have a genome standard. It does not have a genome reference, in which case you have to put all of them together in kind of a logical sequence, but that means the logic can also kind of break down, and it is a kind of an iterative process of fixing. So I'll give you an example, right? I think I've used this example before. Imagine you had a jigsaw puzzle with all the pieces, but you had a picture of what you were going to make. So you could use that picture as a reference to put the pieces of the jigsaw puzzle together. If, on the other hand, there were 1,000 pieces all sitting around, and you had no picture that you could refer to. You would have to do a lot of trial and error to see that the pieces fit, and then for every time, so it's a combinatorially more challenging, is a more challenging process. Okay, so in NGS, millions of such fragments, so millions of Sanger sequencing experiments happen at the same time, parallelly, which all happen at the same time, and then each of these sequences that you get can be put together, and then you reconstruct, a large sequence. So genome-wide sequences for relatively now, however, the technology has reached a point where genome-wide sequences for a small species is possible in a relatively speaking shorter time, right? You could basically, if you did all the sample prep, which probably took 10 days, you could then load all those into our Illumina sequencing uh, uh, instrumentation, and you could get all the data related to a genome within the over the weekend. Okay, so it's that fast. And that is short read sequencing, and then now you have long read sequencing, and things like that. Okay, so why do we need high throughput? Why the kind of one system has to feed into the other? Because information is becoming available at a genome, right? You want to see an expression pattern of certain genes in a patient from a breast cancer survivor, or somebody suffering from breast cancer versus somebody who does not have. So how do you do that? You have to do that at kind of at a very high level. You can't do it one sequence at a time, and therefore, also, unlike the Sanger sequencing, which in the 70s was the absolute gold standard. Now information is becoming available at a genomic level. It'll take about several centuries to do all of this at one nucleotide at a time. Therefore, you have to have a high throughput methodology. And then you also have to have the genome processing is getting faster and cheaper. And you want faster and cheaper because, again, the information that is becoming available is in really large quantities. Okay, no questions, so we'll keep going. However, like if you have, so the way our center charges money is not to talk about the money so much as to talk about the relative scales. And you'd probably, if you had 100 samples, it would probably be about somewhere between 15 to 20,000. Okay, so just because of the nature of the processing and things like that. So what are the advantages of next generation sequencing? It's a cheaper alternative than Sanger sequencing. Now, if you say, well, a Sanger sequencing costs $15, this costs $15,000, but if you took one Sanger sequencing and multiplied by the number of Sanger sequencing that occur in parallel, it would be several million, right? So it's cheaper, and you could do the whole small genome sequencing, can be done in a day. You can find out things about genes, gene expression patterns, expression profiles, whether the presence, absence, the kind of breakdown, the any kind of changes in the regulatory elements associated with disease. And then you can also identify very quickly disease-causing mutations for diagnoses of disease. The expression patterns can also be used for this. And RNA-seq, which is to this point was the more popular because it was really kind of a differential gene expression of two sets of compared sequences. Can is basically the foundations of what is called transcriptomics, because it's the mRNA which is being looked at, not the original DNA. But we learned last time that using reverse transcriptase, you start with the mRNA, but you still convert it into complementary DNA. What are the disadvantages? The disadvantages are obviously the cost, right? And so we are currently interviewing some people, and for a research assistant position. The thing about that is, the people who are working in this field will, in a lab which actually does next generation sequencing, have an advantage, because they have the advantage of basically handling the sampling prep. They have experience with the instrumentation. The only problem is that somebody else may or may not be better, or would be a better research assistant. They do not check those boxes and therefore are at a disadvantage. And the reason is because these instrumentations are very expensive. Most labs do not have them, and that is why a center like ours exists, because in a centralized location we can provide service to everybody rather than reinventing the wheel and every lab bringing their own information. Okay, well, remember when I talked about ensemble. Whenever you look at something at an ensemble level, there are errors, right? So there are errors in sequencing. And then I don't know whether in your undergraduate you learned about what is called error analysis, where you learn using differentiation, the notion of error propagation, right? So, for example, how many of these reads, and we look at what a read is, how many of these smaller, let's say, for example, each Sanger sequencing, and you have several million of them happening at the same time, and we look at this in kind of from a bioinformatics standpoint in greater detail. Once you do this, you'll realize that, let's say there was one error in a 50 nucleotide long sequence, right? And let's say there was one error in every one of these. You could not possibly live with 100 million errors, assuming there was one error in every one. So there are errors, and these errors have to be understood, and some biostatistics has to be done in order to ensure that the errors are minimized. Okay, and for that we have something called as called a quality control. Data analysis and visualization can be a major problem, okay, because again, you're looking at large amounts of information, and we looked at the microarray. If you remember last time, it's like, well, is it sharp green to show that hybridization has really occurred, and it's upregulated, and sharp red where it's down up regulated, and sharp green where it's down regulated, or it's less sharp where hybridization is kind of partial, right? Things like that have to be taken into account. Except now what we are doing is we are doing it in huge batches, right? Several million at the same time, and then you literally take a picture, and I'll show you what that means. At every time one set of nucleotides is added, and when you think about what am I talking about nucleotide being added? Think of it like the Sanger sequencing. Go like, for example, each complementary sequence is added until you get the dideoxynucleotide goes in a complementary way, and then the chain terminates, and then you get one fragment. So many such fragments have to then be put together. Okay, data storage becomes a challenge, right? So, for example, we have in our analysis system, we have uh, we actually cannot store it on the hard drive of the computer. We have an external 5 to 10 terabyte hard drive. The data is there, and then the software reads the data from the 10 terabyte hard drive, right? And so sometimes the drive might fail, which has happened in the past. And then very often, what happened as part of our services, we have actually contracted with the high performance computing center, and we have purchased 100 terabytes of storage space. Now, 100. There used to be a time when Windows came out with its first PCs. Your hard drive consisted of 1 gigabyte. Now you would typically get 1 or 2 terabytes, right? So when you have generate enough data that over time fills up a 100 terabyte hard drive, and then the HPCC people complain and say, hey, you got to delete some of the data or do something about it or zip it or unzip it or archive it in some form. That is when it becomes a really big challenge. Okay, and so data storage is becoming a problem, and things like cloud services and things like that are one option, but eventually you uploading to the cloud will take a long time. Storage in the cloud takes a really long time. So you have all those problems also. Now, there are many different methodologies by which you can. So we're just looking at the basic breakdown of the thing, and this is not an informatics question per se. This is more for the people who are doing sample prep, but I think it's a good idea to get a good look at this. So, in the first, you have to create a template, And each template is, so you have your sequence of maybe a genome level DNA sequences. They're extracted, they're processed, etc. And now what you're doing is you're going to create those target or the templates, right? As for example, in the Sanger sequencing, somebody says, "Hey, can you sequence this particular set of nucleotides for me?" These are then your targets, right? Your target, and then you kind of create the complementary sequence. So once you can build up the complementary, you know what the original sequence was, what the target sequence was. Now you have to build several billion of these if necessary targets. So you have your genomic DNA, or if this is transcriptomics, then you apply reverse transcriptase on the mRNA and you get your cDNA. And then you go through what is called a library prep, where you use kind of a combination of ultrasonication or chemical methods to break that DNA into smaller fragments. Again, this is purely random, right? You don't really get to control it, but there are ways in which you can control it. And then you kind of put an adapter, with using the ligase, you have adapter ligase, and then this is another methodology which uses beads, okay, which uses beads. But we use in Illumina, we use this other method. So basically, what you do is you embed each of these samples on the substrate, and then what you do is you amplify this signal, okay? And this amplification is called as bridge amplification. And there is a video coming up which tells you how it is specifically done in a way that actually it was a video created by Illumina. It'll explain much better than I can, okay? And then what you do is there are two different methods in which basically, so you see that this is your let's say this is the original thing that came from the sample, right? The sample. And then this is similarly, that's the one that comes from a sample. And just like a Sanger sequence, you start building your complementary sequences. And what happens in the Illumina methodology is that you build your complementary sequences. Each of these is fluorescently labeled. So if you say, oh, a T got complementarily bound or hybridized with an A, it has a certain color. And then an A with a T has a certain color, and a T with a has a certain color, and a G with a C has a certain color. So just from that color coding at each addition of each of these of your complementary sequence in a particular location where you know where that sample is, you know exactly which one just from the color code you can tell exactly which of these sequences are added. Now in the ion torrent methodology, you have the same sequence, but as your complementary sequences are added, the pH change is, you register the pH change, and the pH change of that system very specifically reflects which complementary nucleotide has been added, which then tells you what the original sequence is. And then that information, each of this data, is sent for data analysis, okay, which is where we as bioinformaticists come in. And I will actually show you the Illumina process. Okay, the Illumina process including bridge amplification. And let's see if this works. And let me. I don't know whether you'll be able to hear it, so I'm trying to see how the sound will work. Desktop audio. We'll see. We'll see.
The Illumina sequencing workflow.
I'm going to pause it. Can you guys hear? Yes, no? You can just let me know in chat. Can you hear? Can you hear? Okay. Thank you, Ayman. All right, let's go. Keep going. And if not, then you can see the video for yourself when you get a chance. And also, more importantly, you can see the. Uh, let's see if I can make this bigger.
is composed of 4 basic steps. Sample.
Let's see if we can. So you can read the closed captioning
Prep,
also.
cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
So just to interrupt, that's a flow cell, and on that there are oligonucleotides kind of already on there. And so for you ligate or attach to the sample the complementary sequences. So this way, at least you will first get that initial attachment, right? And you know exactly how those are, so you know exactly, and so that's all part of the sample prep.
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away.
Let me interrupt and take a little bit back, okay? And so what happens is you will see that every time over the entire flow cell, you're taking a picture, right? And at a given time, you'll have one nucleotide associated with one nucleotide gets adds on, right? And a picture is taken. And that picture will tell you based on the fluorescent, because each of these are they fluoresce at different colors, which nucleotide got added on. And then you will see, then you have the next phase where you have the next set of, and so you can see these, so you get one picture of the entire thing at a given point in time, and then as the nucleotides get added, as the nucleotides get added, you'll see that the picture changes one step after the other after the other. Okay, so we'll see that again.
Massively parallel process. This image represents a small fraction of
See
the flow
that?
cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
So you have now kind of an idea, like a very nice graphical way of looking at what really goes on, and then when you actually see this in real time, you'll actually get a sense of it. So what happens is once this the point in this case, other than the Sanger like in the Sanger sequencing, the point of the complementary sequence is only to ensure is the point of the complementary sequence is only to ensure that you have the data and the data is in the right order. Okay, so let's keep going. So again, let's now just repeat over what we did. So one step is you start out with a reference DNA sequence, could be an entire genome of a small species, could be the entire chromosome. That's the reference sequence. And then what happens is that the information that the samples that you're given are broken into approximately even size, and this is through enzymatic methods of sonication. This is part of the sample prep. Then to both ends of your DNA fragments via the enzyme ligase, an adapter approximately 50 base pair sequences is attached, and then you use a polymerase chain reaction to make multi-copies of these fragments and denature the fragment into a single strand. All of this is a sample prep before loading it onto your, that's why they talk about clusters. So your PCR is used to create the clusters to make multiple copies and then denature fragment into single strands. Okay, very important. Now, that is the sample prep. Then you wash these single strands over beads. Now, flow cell is the Illumina methodology. Other companies can use something like beads where you have these things hanging off of beads as opposed to that glass substrate. A hybridization occurs when adapters are complementary to the primer, so you're attaching these, and the polymerase and nucleotides are then added to generate double-stranded DNA. But when the double-stranded, so here's the difference is here's the difference between our Sanger and here's, there you don't use gel electrophoresis, okay? You use the methodology that was described in the video. The non-tethered strand is washed away, so you can do a forward and a reverse way of doing it. And but in order to make sure that the signal is really high, you need something called as bridge amplification, right? And so we already seen the bridge amplification. So these are the pictures which actually are just a representation of the movies, and we can kind of go there. And therefore, this one cluster at one point here, which started with a small cluster PCR amplified, gives you a big, so there is a whole region, and then that'll ensure that the light is really strong. But again, this also results in a lot of errors. Okay, let's see. Somebody said something in the chat. Oh, so first, Ayman, you said yes, the sound was working, and then I get the thing that the sound was not working. But here's what I'll do. You have the link to the video. you know, you can always take a look at it. You can always take a look at it. Yeah, there is an issue here with pushing the sound and settings and things like that. So I'm not quite sure how that works. But as I said, uh, you can go and take a look at the video because the link is in the link in the slide that I sent you. Okay, so let's keep going then. Once large clusters of a single DNA with an adapter are attached to the primer at one end, you form. Then you introduce the primers that are complementary because now you, just like the Sanger sequencing, you have a free floating adapter. And the question is, your flow cell already knows exactly what's placed in each place. And the sample prep, they have very specific reagents, and those reagents are know exactly what is going to be on the flow cell. Therefore, there is all of this is very proprietary. So you have reverse termination is same as DDATP allows the adding of only one nucleotide at a time, and every time you take a thing, there is a picture taken. This is again several instances of Sanger sequencing. However, we do not use the gel electrophoresis because you're dealing with so many millions of these at the same time. So at the addition of either your picture is taken, the dyes are differently colored, which tells you that what nucleotide is added, and that tells you what the complementarity is, right? Now, all of this that was shown was in the video that we went through the video. Go to the video again more than once if necessary, and all of this will make a lot of sense. So I've kind of rushed through it because it explains it in a really nice way. So what happens in this case, you can see, for example, in and this picture is separated into different things, and that is one is, let's say, for example, the red means a T was added. So you see when you wash it over, right, only places where there was an A, you'll see a red light. Then let's assume that when you add a green, when you that means it'll add only in places where there was a G. Okay, here if this blue is for an A, which means it'll only be added when there is a T. And similarly, you have ACTG and different colors, and then you create a composite. That's one cycle of adding of nucleotides. Now, each of these in each of these positions, depending upon where your sample was situated, these colors will tell you for that particular sample what the sequence is. And so you get several hundreds of these. Now imagine, now as your phones have gotten better, you can, every time you take a photograph, it's 2 megabytes of data, 2 to 3 megabytes of data, depending upon what kind of resolution you have in your camera, right? Now imagine you have hundreds and thousands of these being taken, right, for the entire thing. So there immediately you'll see that uh, why the memory storage, data storage is such a big issue. So once again, I apologize. When Ayman initially said yes, I thought he said that you could hear, but if one thing I could do is, you have the link in the PowerPoint that I send you. You can take 5 minutes, watch the video, come back in 5 minutes, and then we'll continue. Okay. So I'll just halt this for a few seconds and then watch the video and continue, because I don't want to kind of had these 3 slides of explanation which I glossed over, assuming you knew what was in the movie. So I'm just going to halt, and then you can go from there. Okay, so go take a look at the video. You have the slide presentation, and then we'll continue. And we'll take a usual break anyway, so don't worry too much about it. Yes, I'm still teaching. I just gave them a break. So to answer your question, Michaela, Michaela's question is, it mentioned that the time of the cycle controls the length of the read that is desired. Are there times when you want longer reads or shorter reads? Okay, so this is a question that goes beyond. Typically, you can't really fine tune the instrumentation, at least the way I understand it, so you can have kind of any read that you want. Okay. At the same time, the reads are this the sample prep is basically the length of your read is really the length of the nucleotide you get during a sample prep. And then when you read it and you basically when you sequence that read is when you put those pieces together. The problem is the read lengths are uh, within a certain range. Okay, that's why I said the question was your question goes a lot beyond, like, so for example, you could just say the yeah, your read can be long or short, but you can't like raise it to infinite, right? So in a short read sequencing methodology, the system breaks down once you have, at best you can have a sequence of maybe somewhere between 70 to 100. Okay, it doesn't go beyond that, sometimes 120, but you can't like control it to the point where you can say I'm only going to get lengths of 70. The point about the read is you will get a particular sample length, but your read cannot be more than the sample. That's the point. You can increase the cycle length so that you can get more nucleotides get added to make sure that the sample is completely represented. Okay, that's the point. You can until that sample is completely represented. That means a complementary. On the other hand, you see that in the video they had it. Okay, they set it up and then it kind of just washed away. Set it up and washed away. You can do that. It just gives you a larger number of errors. Now, the other reason I said that your question goes a lot beyond is because you're talking about short reads versus long read. Like short read sequencing, somewhere between 70 to 120 length versus long read sequencing, which is somewhere, like 120, like 1200 or 1300. Okay, that's a whole different question. But you cannot control it. Let's say your sample length is 150, right? You can increase the time of the cycle till the cows come home. So then the next question that you might have, Michaela, and everybody else, is what happens if you um, but if the length of the cycle means that the read can only be this long, right? Let's say your sample had 120 nucleotides, right? But the read is just 70. Well, what happened to the rest of the sample, right? Is the rest of the sample not going to be represented? Are you going to get a misrepresentation? The reason why this issue is taken care of is because there is no cutting point in your big long strand of DNA. There is no cutting point. First of all, when you come up with a sample, DNA is small enough, it's microscopic, nanoscopic enough that you're going to get several 100,000 different strands of the exact same DNA. That is one. Secondly, the cuts are random. which means that you can say, well, I ran my cycle since my read length was exactly 120, or give or take, but my one sample was cut because the cutting is completely random. One cycle was cut, nucleotide was actually 200, the other one was 60, the other one was 50, another one was 500, right? So the cycle controls the length of the read, but what happens is, because the cutting is random, okay, because the cutting of it is completely random, eventually with enough reads you're going to get a lot of overlap. So you'll make sure that, well, that entire genome or that chromosome or whatever you wanted to represent is covered, just because of the overlap. And then we look at a thing which says that this is a uh, it's kind of well represented versus poorly represented, and that also is kind of an experimental artifact. Does that even come close to answering your question? I went about it because, I mean, the kinds of questions that you asked had something a lot more than beyond the context of what I was trying to answer. So I'm not sure whether that answered the question. And you can unmute and ask again, so we can look at this a little more in depth. Okay, thank you. So once again, when the sample cutting occurs, it occurs at random. Okay, but if you keep your cycle the same, the cycle means that, oh, within this period of time, you're going to have these many nucleotides added. For the most part, you're give or take, you're going to have the same length. Are they? But let's say we just say, well, if that's the case, why not have as just prolong the cycle until the whole thing stops and every nucleotide length has found its complement, and once it's found its complement, it's just going to sit around doing nothing. Okay, that allows a lot of error to get added. But again, from an experimental standpoint, long read sequencing, like whether you have something called as PacBio, which we have or recently acquired, and another instrument called Oxford Nanopore, which is kind of the background science is different. You could get up to 150,000 nucleotides read length. So now again, because we're talking about it, and we won't talk about, and I'm glad you asked this question, Michaela, because we're not going to discuss this any longer. I mean, in terms of a long read sequencing, the idea is, let's say you have a gene. Okay, let's kind of scale it down to just one gene, just a functional part, just the open reading frame, and you cut it into various parts and you chop it, right? Random parts, so you can actually kind of, like a jigsaw puzzle, you can recreate the entire gene. Let's say that this gene is 2.5 million nucleotides long, okay? And let's say you have short reads, 100, 120, etc. You're going to miss some middle portions, you're going to miss some middle portions, which means it's not going to be a proper representative. Secondly, you might actually get a match of the read to the gene, let's say in the original genome, that might be kind of random. So it's like, okay, this matches this particular gene, and I'll explain what that is. This matches this particular gene, but really, it's a random match. You remember how we talked about when you did blast searches, your E value is a quantification of the randomness of that match. So the smaller the sequence, the greater the chance of randomness. If you have a big long sequence of 100,000 nucleotides or 25,000 nucleotides, it matches a gene which is 1.2.5 million, you know that region is properly, that there cannot be another region like that in that gene unless there's some sort of a replication. But if you have smaller pieces, because the smaller pieces tend to repeat a lot more often, and therefore, okay, I'll look at it from the context of a protein. Actually, over time, the three or four students have been working on kind of related projects, and we began to look. Let's say you take a protein sequence, okay, just a protein sequence, full functional protein sequence, 350 nucleotides, which is amino acids, which is standard. And we actually did a study. Actually, we should be publishing this sometime soon. Is that there was a what are the repeat regions in a protein, right? So let's say I have AAA, and then that AAA, is it repeated in other parts of the protein? How many times is it repeated? right? Then you have ACTG, right? I mean, I'm talking about not ACTG, I'm talking about glycine and threonine and alanine, and right. So the protein part, are they reproduced? So you see, the longer the sequence, the longer the region. Is it repeated in other parts of the protein? And I'm not talking about dimers and multimers. I'm just talking about in that single monomeric unit of that protein. Uh, is it? Yes. But you know that it never goes over 7. The largest the largest the region of a match, or that of amino acids that can be repeated within that same protein is 7. It does not go beyond 7. We've tried this for several thousand different proteins. I don't know what the kind of the evolutionary consequences are, the consequences of kind of the maintaining that variability and the non-repeat region. I don't think but I've never seen anything where you have a region of 8. On the other hand, for example, here's a region and it's an amino acid 3. Just again, contextualize your big sequence from that point. It's like, oh, there's a region 3 here, and there is a region of 3 here, another part, a region of 3 here. So which part of the gene does it really come from, right? Now that becomes random. If on the other hand I had a 20 amino acid length of amino acid, and it would probably match just to one place, and I would say, yeah, that comes from that part of the gene. I'm just giving you an example of a protein just to make that point. So hope that helps. So long read sequencing are designed to remove that randomness. Okay, so once again, this cycle, this little piece that you got, that aligned in a complementary way to a sequence is called a read. Now, all that strum and drang was to come to the bioinformatics part of it. This is not bioinformatics part. An experimentalist will explain what I just did in a lot better way. Okay, so how do you assemble the reads into a coherent sequence? So let's say here is a read. Here again, remember, so this entire region, see, that's one of the reasons why you could, if you have a longer cycle, you could have a longer read, and a shorter cycle, you could have a shorter read. But the idea is, there is, because of the randomness of this during sonication or enzymatic breaking, it's so random that you're always going to get sufficient overlap. You might also not, but you're, for the most part, is designed to give you sufficient overlap. So see, in an experiment, you got these random breaks, right? See the random breaks? And you align them. Now, here's the beauty of next generation sequencing. You come back to the first principles of the aligning process. You come back to the first principles of the aligning process, just like you would do in a blast search, or just like we did in our first study, where we looked at the, just as we did in our, when we did single sequence alignment and blast and binary, like pairwise alignment and things like that, right? The same methodologies that you used in the basic sequences are now being used at the genomic level. And this is a consensus sequence. So let's say, for example, this came from the genome, right? And so what happens is, you're going to see all these overlaps, and this overlap, and a software will typically do that, will actually do that kind of alignment and say, oh, these are all the reads, but looks like all of these align to this particular sequence. And then how many of these reads form an alignment with this part of the sequence actually gets a count. And when it comes to RNA sequencing, so let's say, for example, you would say this is 1, 2, 3, 4, 5, 6, 7, 8. All of these, let's say, so this read count for that particular region and or aligns to this particular gene would be a count of 8. Okay, so you will see, however, that because of whatever reason, you will see that there are some areas that are not aligned. Okay. Now, what you will see is, so you have the multiple copies of a genome, and from there you randomly cut them, and then you kind of went to the sequencing, and you got these reads, right? These are the regions of the reads, and then you're aligning them against each other. So what happens in this case, probabilistically, looks like you have, based on the overlaps, etc., you have come, it looks like it's well covered, right? It's well covered. That every part of this, because of the way these are organized and aligned, they're covered. That's called a high coverage region. And sometimes you will also have low coverage for whatever experimental artifact or any issue or sampling problems, etc. You will have low coverage. In which case you might say that here, though it feels like, oh, it is looking at this part of the, you might say that because you do not have enough of a sampling, it's almost like, there's a greater level of uncertainty in that identification. Okay, so that's what it is. And again, then you look at the data. Obviously the notepad style is antiquated because you're not really going to have a lot of, but you can open a FASTQ file in a notepad format, and I'll show you that soon. You can store it in Excel spreadsheets once again, but the data is too large. It's going to take a lot of space, but a database with a little more of a, whose architecture is designed to uh, is designed to store, is a better way of storing. But even though you have databases or Excel, or basically your FASTQ file is in a notepad style of information, it is a lot, it is a lot, it is better organized. Now we have the data in what is called the FASTQ format. Okay, the FASTQ format. And so now remember, this is fast that we looked at when we looked at sequences, and I started by teaching you what the real format of a FASTA is. This is what is called a FASTQ format. So how do you send information related to a particular, how do you send information related to a particular gene? You send it in what is called the FASTQ format. Okay, so let's look at the FASTQ format. So a FASTQ format, like, remember the FASTA file, just to kind of give you some sort of a reference. The FASTA file, the title of that particular, you can, first of all, you can have multiple sequences, billions of them in the same file, doesn't matter. The FASTA file is something file name.fasta, or file name.fsa, or file name.fa, or file name.fas. Okay, there are different extensions to these files. Now, and remember the FASTA file, it started with a greater than sign, and then you could put enough information about that protein or about that gene or about that DNA nucleotide sequence or mRNA sequence, and it could go on forever, right? You didn't really, there was no limit. On the other hand, in a proper FASTA formatted file, you will see that the protein information and the gene sequence could not go beyond 80 characters, and you had to go to the next line. Of course, there's no limit to the number of lines. There's no limit to the number of lines that you might particularly have, because for example, you could have an entire chromosome, and you can go to GenBank, an entire chromosome will list it. It'll go into 50,000 lines, doesn't matter, but it does not go above 80 characters, because a software that reads a FASTA formatted file will not read beyond 80 characters, right? If there are fewer than 80 characters, it'll go to the next line. If there are more than 80 characters, it'll go up to 80 characters, ignore the rest of the file, and rest of the line, whatever's in the rest of the line, and then go to the next line. Okay. Now, a FASTQ formatted file is in 4 lines. Okay. Information about that sequence is on the first line. And rather than start with a greater than sign, as you can see here, it starts with an at sign. Okay, at sign. So sequence ID. Now here you'll have all kinds of information related to that particular sequence. It'll tell you about, again, just like that, it'll tell you under what conditions something was determined. There are these things called cigar scores, which we'll talk about. More information about the quality control. Anything that you could put here is fine. Now, here then is the read. Now I don't know how much this is. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then 10 will be 20, 30, 40, 50. Okay, this is a truncated system. It could be more, it could be less, but once again, now we know something a little bit more about the reads. So this is your read. This is your read sequence, right? Depending upon the cycle, when it stopped, that little part of the read was kind of washed away. It was washed away because you already know uh, what the fluorescent lights symbolize, right? So that cycle was already complete, and then you can also redo it on a paired way. Okay, then the next line is a plus sign, and that plus sign purely just separates the read nucleotide sequence from these symbols. Now, what are these symbols? These symbols are basically the quality. Remember, when we talk about an ensemble, and this is really, really, really important, when you talk about an ensemble determination, everything is error prone. And when I say error prone, it means that there is an error in how the new. Now you might say, but why should there be an error? Isn't complementarity sacrosanct? Is it like if it's supposed to be complementary and A will only go with the T? Yes, it is. But sometimes what happens is, as each of these gets added, maybe your cluster is not has the bridge amplification didn't amplify that cluster enough. That particular sample is weak. The signal that comes off it is weak. Something that looks like a yellow is necessarily not yellow. Maybe it's light green, right? And therefore and then what you have a sensor, because as each of these layers of lights happen, right, based on the fluorescent lights, it gets recorded. Now, sometimes the recording is not necessarily correct. So there are errors at various levels. Now, what this gives you is the error in each of these positions. Okay, each of these positions has an error associated with it. So based upon these symbols, it tells you. Now, you might say, why not just call it 0 to 9? 0 being 100% chance that that is a correct nucleotide, and 9 being a less chance. Well, that's because the variations, the kind of the subtleties in how those are a lot different. And that's why I included this particular. So this is the scale. This side means stronger probability that it is right. On this end, which ends in the tilde sign, is where it's the weakest. So these symbols, numbers, capital letters, good chance that the nucleotide in place is correct. And remember something, we are doing this for every nucleotide. We are doing this for every nucleotide, okay? And that is very, 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 very important. Very, 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 very important. We are doing this for every nucleotide. All right. And then in terms of the potential parsing of the files, is not really an error. For example, you see an and sign. There might be some confusion between the and, which signifies the beginning of a FASTQ data information, versus if you had an and sign here. It's not likely to be a problem. Another thing that is a possible problem is a plus sign, but the FASTQ processing instances of information, they know exactly how this is supposed to work. Okay, now go and take a 5-minute break, and we will deal with other things like Fred quality score and things like that. Okay, and because I need to assemble some more information. Okay. Okay, let us continue. And before we do anything else, I wanted to kind of explain to you what do we mean by kind of the informatics, and then I will show you some real life data. You don't have to do much with it other than just understand foundationally, okay? And the two things that we are going to work on is one is one is RNA-seq. How is it done? RNA, sorry, I think I can do a better job with that. RNA-seq. Okay. Now, let's say we have a, let's say we have, this is a gene. Okay, this is a gene. And let's say that we have various samples. Okay, actually, let's say we have two genes. This is just so you understand conceptually what is going on. Okay, let's say our entire genome consists of two genes. Okay, I know, hard to believe, but let's say it's done, right? And so what we did was, as we went through this process, we went through random sampling, right? We cut randomly, we loaded all the samples, properly ligated and adapted and all of that stuff onto our flow cells. And then we went through the process of generating reads. And let's say we generated a bunch of reads, right? Read, 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 read. Okay, and similarly, we can say we had a read, read. And again, you stop me if some is not clear, but I'm trying to make this about as clear as possible. Okay, now let's say that in these two genes, we found these reads, and those reads, so we have two things. We have red genes, and we have blue genes. Okay, genes as in GENES. And let's say that this is for, which means that these reads, the little pieces of nucleotide that we got from our FASTQ files, were aligned nicely with this gene and nothing else. Okay, so that's basically what it is. A piece of read, you send it through an alignment process through your entire genome. That's why that's a reference genome, and it aligned to the gene. So we said the red is represented by 1, 2, 3, 4, 5. Okay, so the red score is 5. Okay, that's it. That means 5 reads. Now it could be a lot more. And again, there's a normalization process. There's something called RPKM. We'll worry about all of that later. I'm just trying to explain to you conceptually. Okay, that 5 of these reads during the bioinformatics analysis, using the pairwise alignment, whatever methodology, 5 of those reads out of 25 trillion. So that's not what we're interested in. We're only interested in this. And then 6 of these reads, 1, 2, 3, 4, 5, 6, aligned with the blue. Aligned with the blue. Okay? Now, this is, let's say, for a healthy individual. And this is what is this idea of, and let's say there were some other samples related to a disease individual, and I'm going to say that for that particular disease individual, we had so many reads aligned. Okay, so now what you see here is that initially for a healthy, we counted 5 reads. So we said the count for that gene is 5. Okay, that means that particular gene is expressed, that expression number is 5. And then for the red gene, for the blue gene, the expression number is 6. Then we look at that for the disease individual, and we find that the expression number, the expression, and the expression is quantified by the number of reads that become available. Remember, because if that gene is present in a large preponderance of amount, you're going to have more samples associated with that, right? And therefore, this number, in a very kind of a vague sense, is a representation of that how much of that gene is present in that healthy individual. Now, when we go to a diseased individual, we find that the number of genes was 3. That the number of gene that sorry that the read count was 3. And then we said for the blue gene, the read count was 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. In a diseased individual, that means that gene expressed or was present, right? Remember, we have a genome and everything, but our expression levels differ. So let's say, for example, this was a liver cancer tissue, and in the control, we found that the red gene was 5, and the blue gene was 6, which means, again, the number just represents the level of expression, right? That's why this can only be done in a differential way. But in a diseased individual, using the same methodology, we say that because the number of reads corresponding to this gene, and I'm using just simple numbers so you can understand, was a lot more in the diseased, and then in this it was a lot fewer. So what we end up doing is we end up taking the ratio of disease to healthy and disease. So in this case, 5 divided by 3 is let's see, 3 times 1 is 3, and so we call that the differential gene expression, or this is called as the ratio. Okay, that ratio of 5 divided by 3 is 1.6. 1.6 for the red gene, and then 6 divided by 13. Okay, 6 divided by 13 is 0.5, 0.46, but let's say 0.5. So now what happens is you say that because there are more reads, because the more the amount of gene, the more the sample that comes from the gene, the more the number of reads that come out of the gene. So in an experiment that is properly controlled, healthy in the sense that all other conditions being the same, for some reason the healthy to disease ratio is 5:3, or the ratio is 1.6, and then the healthy is 0.5. Okay, healthy to disease is 0.5, which means you say that in the healthy, the diseased, the red gene is down-regulated, right? Down-regulated, and in the diseased individual, this particular blue-colored gene is upregulated. Okay, so now from a statistical standpoint, you say you do the ratio of the two. Okay, you do the ratio of the two, and you say, well, this is 1.6, this ratio is 1.6. Now, what we are looking for is what is called a twofold. Twofold. That means the ratio should either be 2 or higher, which means if it's double or higher, or half or lower. So if it's double or higher, you say it's statistically significantly upregulated. If it's 2 or if it's 0.5 or lower, now this does not meet the criterion because does not meet the twofold change. So we say, well, 5, then in this case it's 5, in this case it's 3. It might not be statistically significant, because remember something, the point of doing something like this is you say, I have double this amount, right? Or triple this amount. I need to have double the number of genes expressed, which is represented by double the number of reads, and that's why it's expressed. However, 1.6, you could use it. You could just say my cutoff is greater than 1.5 or lower than 0.3, but in this case, you say, no, I don't, okay? Because we're looking, our criterion is a twofold change for upright. So you say, well, it could be, but I need some statistical significance before I point to that gene and say that red gene is responsible for, or something related to the red gene is responsible for that particular disease. And you want to be sure, statistically sure, that, so you call twofold. Now, on this guide, you might say, well, this is marginal, but looks like the disease, there is some down regulation or up regulation in the gene, or down regulation when it's compared to the healthy gene, and you say that. And this is basically the foundation of RNA-seq. Okay, this is the foundation. This is the foundation of RNA-seq. This is what we are measuring. Now, in terms of what we are measuring, let's go to a real life example. I've got two of these open, so let me just shut down one. Let me just so what happens here is we have looked at, and again, there is some propriety about this thing, so don't worry about it. Don't worry about it too much. So what we here is we are looking for microRNAs. Okay, this is microRNAs. So you see that this is day 1, okay, day 1, day 2, day 3. So you'll see that this particular microRNA had not changed, right? That means it's 0. The change is 0. On the other hand, this RNA, this ACT1 gene, on day 1, whenever it was measured, let's say they were measuring the expression of this gene, it was 822. Then when it was on day 2, it went to 0. And just because the ratio, because something over 0 is a non-zero number, it will give you an infinity. Therefore, you try to make that denominator non-zero just so you can get actually a small number. Alternatively, if you look at the behavior, so this is what I'm saying, right? This is like the red gene. What is the expression level on one day? So here we're not comparing healthy versus disease. We are comparing the same individual. How does that gene express? Which means that, let's say, give or take, this is a normalized number. It's not an actual real raw number. But you can say that 822, this gene was 822 reads were associated with that particular gene when you looked at the samples for day 1, nothing for day 2, and nothing for day 3. The Act B, on the other hand, you say that gene had no reads associated with that particular gene on day 1, but it skyrocketed on day 2 and nothing on day 3. So this is what was supposed to be of a particular condition. Let's not worry because it's kind of proprietary. We haven't published this data yet. You don't know exactly what that number is, right? So these are the kind of numbers. Alternatively, you see that Act A1, you'll see that nothing on day 1, 16.5, which means there is some expression by the number of reads. And again, these experiments were conducted under the exact same conditions. So if there are no reads, which means that is not expressed, okay? And in day 3, similarly here. So you see this ACP1, this gene, this gene, this gene, something happens to them on day 2, nothing happens on day 3. If you go here to this AP3B1 gene, you'll see that on day 1 and day 2 there's nothing. So here it's being compared with itself day by day by day, and you can see all these numbers, and that this is just a kind of a color code to you see that the blue means nothing, yellow means, just so you can get a snapshot picture of this. Okay, So you will see you can even have numbers like 1221. So here you have no upregulation, then you have no, then you have some upregulation compared of that particular gene. And how is this measured? Just by the number of reads. So if your sample on day one, if your sample on day one had many sampled, and therefore so many of those reads mapped to that particular gene, in this case the back BCAP 31, then on the third day, so this was almost like a longitudinal study in a particular condition. How does these expression levels change day by day by day? Okay, so that is, so again, and then the association would be, it's a very clever study. Then the association would be how would those genes work. So this is a typical RNA-seq experiment. Okay, this is a typical RNA-seq experiment. Any questions? Anything weird? I'm trying to explain again very conceptually, using very simple terms, very simple, just blue gene, green, red gene, the number of counts, and the counts, how do they count, is because they do alignment, the pairwise alignment, just like you did in earlier, which used to be the big thing in bioinformatics. You're just aligning against a reference genome. And, oh, these many aligned, I'm going to put a count. And then I'm going to normalize the count. And it's like, well, what happens under the similar conditions for that same gene for somebody who is a patient? Like a cancer tissue versus a normal tissue. And then that's the point of the expression analysis. Oh, so in the basically it just means that the blue means nothing happened, Michaela. Blue means nothing happened. Yellow just means that that you see a non-zero number. Now, if I look back here, there's not much here to say. Let me look in this. Yeah, these are some other calculations I did. It just means there's a non-zero number. But yeah, so again, these are just color coded to say that there's an, because you can see now this has been like several years since we looked at it. Zero means blue. Now, if I had included, and this was just so I can get a snapshot view of this, obviously it just means that I can get a snapshot. Oh no, on the other hand, you see here, what I did was you just use Excel to like color coded within range. So you can see this is 76,000. So up to even this 8,000 is yellow, but this is kind of slightly orange at 31,000 and red. So I just, because the range was pretty big, I was like, for different ranges, I could have done the log of this. For each of these ranges, I had a different color. So it had to be, in order for it to become red, dark orange versus light orange. So to answer your question, it just gave me kind of a snapshot view. For example, here you'll see the numbers now are really high, really high. Okay, but this also tells me that that between this and this, right, if I took a ratio, you can just say it's not more than 2, right? It's not more than 2, because so I'm saying that this is like, almost like a housekeeping gene, like at least these are housekeeping genes. It doesn't, that's why you need that twofold. Like for it to be significant, you say it's got to be more than double or less than half. And in this case, you'll see that these numbers don't change. Though, I mean, these just tell you what the types of colors are. And this is obviously the biggest number. And so now in this case, it goes high, 140,000. Then it comes to 12,000. Then it goes back to 191,000. So we're just looking at gene behavior over time. But again, this is within the same individual, how these gene expression levels change from time to time. So each of these was like, say, like this is subject 6 sample from day 1, subject 6 sample day 2, subject 6 sample day 3. There are other subjects which have 9 and 10 days. So this was just for me to look at it. So you can see then the color codes were like, if it goes above a certain level, it's red, and so now because these numbers are big, you get the orange and the red. But if the rest of these are, even though it is 3,500, it seemed important in the previous case. So you'll see that this is how the profile changes. I can go to another subject uh, where there was some comparison. like for example here, subject 4, I can open this up and you can see that there was some comparison here, right? But the red will show some really. So these are just color code. It doesn't really mean much, but it gives you a snapshot glance. So I just basically took that whole thing and I said based on these numbers, so this person only showed up on day 1 and day 2. So because when you show up only on those many days, that's when it becomes a problem. But I still kind of ranged, like I put some conditional formatting in Excel just to see the, so I could at a glimpse. So if I make this really small, you'll see that I can tell here, if I point to this part, then I'll see that this gene, this RNA something something, it was really high. So 394,000 was versus, so this is really upregulated, right? From day 1 to day 2. And that gives us a picture of the idea. Okay, so yeah, so blue means no change from place to place to place, but blue and yellow doesn't really mean much except when you look at the colors. If there is something, then there is something a little deeper. So red was the highest level of upregulation, and then I changed it, like if it was a down regulated thing. This is just one example. I basically used kind of colors in shades of blue. So this was just for me and for my collaborators, colleagues to get kind of a quick view as to this. And we've done, this is just one example. But again, the idea is if you see a number, that number means that these many reads, not 839 reads. So this is kind of a normalized number compared to everything else in the RPKM value. And then so you can see that there was some expression and that expression decreased on day 2. Now, in this particular study, one of the problems, one of the reasons this particular study suffered is this particular study did not have anything like all the patients didn't show up, from supposed to be from day 0 to day 11, and most of them like didn't show up. A few of them showed up, and there were some women, but only one person did the entire, like all 9 days or 10 days. And some of the women never showed up after day 0. Some of the women showed up after day 3. So what happens is, if we were going to look at some profiles and say, well, okay, here is a profile which is different based on sex, but you really could not do that with any kind of significance because only one woman showed up for all days, right? So after day 4, for example, any result was purely because of one woman, and you would not, you wouldn't look at data like that and say, well, yes, now this is applicable to all women because the sample size, the sampling is really poor. Again, it was the nature of the study, which was a really smart study, but execution, they had problems. And I think this is, how they have in psychologists, they have this notion of surveys. So they self-report, and self-reporting very often is fraught with all kinds of issues. So the self-reporting was the problem. Okay. All right. So this is a typical RNA-seq experiment. Okay. So you're comparing your reads to the genes, and the number of reads, the more the number of reads, that means that gene is expressed better. Okay, but this works a lot nicer if you're comparing. So for example, in the study that you're looking at right now, you are comparing the same individual from day to day to day. How do the expression profiles change? You could also alternatively compare two different individuals, or take many individuals which have a particular phenotype, a particular disease, and many individuals who have not, and kind of take the average of all through. But again, it's always a comparison. Okay, and the reason and again, there are many other things like single cell nucleotide RNA sequencing, single cell sequencing, whole genome sequencing, chip sequencing. I'm just using two as an example because this is kind of an introductory course. You would have to delve deeper into, and there are many. Some are commercial software, some are free software, some software require Linux, and all of these things. Now, using the exact same analogy, okay, using the exact same analogy, let me go and delete all of these things. Oops. Oh. For one fleeting second, I was terrified. I said, oh man, I deleted the entire thing. Okay, so now let's look at the next part, and that is metagenomics, or sometimes called as microbial genomics. Now, in metagenomics, now the idea is the same. Okay, the idea is the same. So now let's just use the same kind of analogy. Let's say now, but now rather let's directly look at the comparisons. So let's say you go to uh, one part of, let's say you go to some, the hill country in Dallas. Let's say you go to an agricultural country, and let's say you get a sample of the soil. Okay, then let's say you go to West Texas, and then you get another sample of the soil. Okay, and then this is what is called amplicon sequencing, where you basically you take the 16S, the 16S, which is a very signature gene for you have the 18S and some other sequencing for fungi, etc., where you amplify the 16S sequence gene, which is a signature representative gene of in bacteria. Okay, so this is Hill Country, Texas, and this is Lubbock, Texas. Now, you can see now again from a sample prep, RNA extraction, DNA extraction, the methodologies are different. Okay, are different. But I'm asking you to kind of go up there in a drone at 5,000 feet and look at this idea of this thing conceptually. Okay, that's what we're interested in. So let's say we find we are looking for bacteria, and this is a 16S, so you amplify it, or you amplify certain what are called hypervariable regions, which are the ones which basically differentiate. And you look at the hypervariable regions and you look at the 16S of, you're not just looking at, the reason it's called metagenomics is you're not just looking at one species or a tissue from an individual or a group of individuals that manifest the same phenotype. You looked at these two soil samples, and in this soil sample you identify the bacterial profile in this. Okay, you amplify the 16S of all the bacteria. You amplify the 16S of all the bacteria in the sample from Hill Country, and you amplify the bacteria from, and so you get a bacterial profile. Now, as I begin to draw this, you will see that there is some similarity. Now, let's say this entire planet had only 5 bacteria. Okay, this, again, keep it simple. And so what you will see here, one is the orange bacterium. Okay, orange bacterium. One is the dark blue bacterium. And let's just keep it simpler. And then one is the green bacterium. So this is almost like there are reeds. They're almost like reeds. So you do the exact same, different methodology, but you're still using reeds. You're using reeds. And now each of these reeds is basically maps or matches, or using pairwise alignment, comes from the green bacterium, the blue bacterium, and the orange bacterium. Okay, so then I have one here. Let's go to blue. And then let's go to the orange, and I have 2 here. So now what happens is the read, just like if a gene is overexpressed, you're going to get more reads because there's more of that gene in that particular sample. And you're going to then say, oh, more reads correspond to greater amount of gene, especially when you do some comparison. Now what happens is you're saying how many bacteria are there. So here I have 3, and here I have 2. Okay, same sample, not same sample in the same kind of sample from 2 different places. This has 2 bacteria in it. This is 1, 2, 3, 4, 5, and this has 4 green bacterium, and 1 of that, which means that the reads now correspond to, because you amplified kind of the bacterial profile, you will say that in the hill country specimen there is a lot more of green bacterium, there is but less of here. The blue bacterium is more in Lubbock than in the hill country soil sample, and then there is 3 and there is 2. Now guess what we are doing? 3 and 2, we're taking the ratio. 2 and 5, we're taking the ratio, and 4 and 1, we're taking the ratio. Now 3 and 2 gives you 1.5, this gives you 2.5, so we say, oh great, that means that is statistically significant large amount of bacteria. And then this is like 2 to 5 is 2.5 divided by 2 is 1 point whatever, and this is 4 times, right? So these are statistically significant. Do you notice a similarity? While the metagenomics is to look at many genomes, right, or many 16S, which are the like kind of the signature genes in bacteria, and you see there's more of one more than the other. And that's basically what we are. And then the rest of the procedure in terms of normalization, etc., is all the same. Okay? Then, so the question is, how do I know what bacteria there are? I don't. So what I do is based on these reads and these sequences, I actually run my process against a database of 3.8 million bacteria. That's the one that I typically use, okay? Depending on where you have 3.8 bacterium, the 16S sequences of these bacteria are there, and each of these reads then align against each of these bacteria, and then the moment you get a hit, like in a RNA-seq, you'll say, oh, it mapped to that gene, so I'm going to put count 1 for that gene, or count 2, depending upon the number of reads. Each of these is mapped to a different bacterium, where I have a kind of an archive of the 16S sequence lengths, and it then identifies the bacterium. And then when you do this comparison, you say, yes, statistically significant more of the blue, and statistically significant more of the green, and there's a little bit more of the orange, but it's really, we will not consider it statistically significant. So do you see the parallels? In one case, of course, you're actually identifying genes. In the other, you're basically identifying entire bacteria. So you take it one step higher, and then you call it the meta. Okay, that's metagenomics, microbial genomics. Experimentally, sample prep, amplification, etc., is different for fungi, but the foundational bioinformatics is still the same. You are in metagenomics, you're taking the sequence, and you know that that particular read corresponds to the 16S, which represents an entire bacterium. On the other hand, in an RNA-seq experiment, it represents a single gene, and therefore it's a quantification of its expression levels. Okay, so now let me show you. Let me just change this a little bit. Oops. So now what we have here is, so I see a map here, right? I see a match here. Let's go back. Now, so what happens here is Now this is like different samples, okay, different samples taken from different places. Okay, and so there's a lot of different samples, and you'll see most of them are, when the number is same and small, it's 0. So on the other hand, you see that this particular bacterium, the ID, and this ID is from the database that I use, is, they've got something in sample 1, then they've got something in sample K, something in sample N, something, but no evidence of those bacteria in the other samples. Now, I gave you the example of soil samples, but this is the feces of hogs, okay, or coyotes or something like that. So it doesn't really matter. You take that thing, you express the, you amplify that DNA, you amplify that gene for that particular bacterium, and then you go through the process of creating reads. You run it in the exact same way in RNA-seq, but rather than map it to the single human genome or dog or cat or rat, depending upon what species you're looking at, here we run it against each of this is the bacterial species. So you will see that here you will have numbers, nothing, 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 numbers, numbers. So each sample is represented very differently. And then we do other things. So for example, we say, okay, that one it's an uncultured bacterium, which means we don't know much about it. But then but however, the database tells you, what family it comes from. Firmicutes, this is not the family. This is like the domain. Then it comes the phylum is from the Clostridia, and the class is Clostridialis. And in some cases there's no subclass, and then the order is Ruminocaceae, and then Aceteinobacterium, and then here is a 16S sequence. Oops, you can't see it. And that is the beginning of the 16S sequence. So again, the point I had for doing all of this was merely to illustrate that at the end of it, you are still comparing. However, the methodological way of identifying the sequences, amplifying the parts that you want, creating the reads, is still exactly identical to how you do it for both of these, right? And that's one of the reasons, if you look at this PowerPoint, you will see that the foundational idea is still basically the same. Any questions? I tried to make this, to kind of do it in a more of a hand wavy way, because I didn't want anybody to get caught in the weeds. But foundationally it's all the same. And sometimes this commercial software that we often use, they will actually tell us, it's like, don't use the metagenomics thing. The RNA-seq is a lot better. And then there are other things that you have to understand from the perspective of the other thing that you have to think of from the perspective of this of understanding some of this is that experimentally you don't have to worry about it, but from a bioinformatics standpoint, it's the same. It just maps to something different. Also understand that everything you learned in terms of pairwise alignment is the same. Now, as you create the criteria for your pairwise alignment, when you look at the criteria, you say, okay, when is it supposed to be considered a match? It's supposed to be considered a match if there are only 2 gaps. If there's more than 2 gaps, please don't consider a match, because that could just be a random alignment. Okay, we're dealing with this at an ensemble level, like millions of these are happening at the same time. Okay. All right. So while we're at it, let me go and show you an actual FASTQ file. Okay, this is an actual FASTQ file. And now remember, this we can go anywhere here. Remember, it starts here. Okay, um, it starts with your @ and that's the ID, and that tells you some other information, experimental information. It might actually tell you like from where to where, in the sequence, what does it map to, all kinds of additional information. And this tells you, I think, information about the primers, the primers associated, because you can see all these numbers are the same. And then here is the here is your read sequence, and you can see the read sequence looks like it goes to about 100 and something. Okay, and then here is the plus sign, and it tells you that all of these are in the F. All of these are, in terms of the quality. So you can see, let's go up here, you see here F. So what is F? Let's go back to our PowerPoint and see, for example, if you see uppercase, it's not bad, F, and then you'll see there are commas and things like that. So you can see here that, okay, so here you have some colons, comma, colons. That means, so remember, for each of these sequences, each of these nucleotides, you will see that, so you can see the quality is pretty good because colon, I think, is good. Colon comes on this side, colon comes towards the left hand side, so it's good. See, for example, you can see right after the 9 is the colon, right? And so that's good. So some of the qualities are actually pretty good. Okay, now let me scroll down a little bit, and you will see that here we have a whole bunch of, this is not known, not known. Okay, N is not known. So something happened with that read, that nothing was found, right? And that's why. So now if you look at this particular file, okay, has got, now we take the number that you have and you divide it by, so this is line number one, and it goes to 429, sorry, 4,296,524. See, so this is one FASTA, and each fast queue. This is the fast queue. There are right now, this particular experiment has 56 samples, so there are 56 fast queue files, and this is because I wanted to open it in my Notepad plus text file, not Notepad, regular Notepad. It wouldn't open, but you can see that this is close to half a million. This has close to half a, well, 4.420, 4 4 million 200, so it's close to, it's somewhere around 5 million, little less than 5, more closer to 4 million. You can see each of these. Okay, so this is a real life experiment as to what you would typically expect. Any questions related to this? Okay, so once again, I showed you that other, to show you how foundationally, conceptually similar metagenomics versus, and again, it's always about comparison. Now, I won't go into whole genome sequencing, but what the whole genome sequencing will do is you sequence the entire genome, and so this is now not about gene expression, but what you do is then you run it against an existing, then you do a comparison of an existing, and what you'll typically get from a whole genome sequencing is the mutations, the SNPs, like, uh, uh, because you're not really amplifying specific genes, you will get, where does one gene, where does it occur? Where does, for example, in a particular gene, do you have, according, when compared to your standard genome reference, do you have is there a SNP? Where is the SNP? How do you identify the SNP? There are these things called SNP arrays and things like that, so you can also say, okay, here is a mutation, here is a mutation, here is a mutation, and that is, you take your entire genome and compare it to your standard genome, and you will basically find that. That's whole genome sequencing. But because I just wanted you to take a look at, like, an actual analysis that we typically do. Now, remember we said, Radhika, no laughing, I'm teaching. So remember how remember how I said that that there is a, when you look at our FASTQ files, you'll see that there is a quality associated with every time a nucleotide is added, right? There is a quality. Now, the time nucleotide is quality, 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 quality. Every time a nucleotide is added, the quality of that is added. So now we actually have to take a look at that and ask ourselves, how is that quality? What is that? What does that quality mean? How is that quality determined? Okay, and the way to quantify something like that is what is called as the Fred quality score. So the Fred quality score measures the correct base in the specific position, and what is the quality, how certain can we be that that new nucleotide that we added is the correct one and is really a representation of the nucleotide that comes from the sample. Now this could be various things. It could be an area where that you didn't see the color too well, your sensor didn't pick up the color too well, or it was, or the color didn't show up at all, and or color just showed up very briefly, and your sensor that is supposed to pick up the different colors and assign the nucleotide didn't do a correct job, wasn't very certain. Again, because this is an ensemble determination, there's millions of these at any given time, and several millions. Now we saw that just one sample, right? And all those samples, all 56 samples, which means each of them has about 4 to 5 to 10 million reads. Each of those was all put on the same experiment. So now you kind of get a sense of the scale and therefore the need to to create this quality control. All right. So take a look at this and you will see that, let's say this is your read. Okay, these are your read, GATTTTTTT. Now, you see this blue line, which is something called as a Fred score of 20. We'll worry about that later. And you will see these lines. You see these vertical lines? You will see these. Okay, so let me do it again. This is the read. Looks like it has this is position number 60 and this is probably position number 100. So it's about 100 nucleotides long. And you will see that there is a blue line, which is basically what is the cutoff between acceptable and non-acceptable, and that is what is called a Fred quality score of 20. Now, in the next panel, in the panel here, I'll tell you how that quality score is controlled. So 20 does just seem some random esoteric number. That's not what it is, okay? Now, you'll see these vertical segments here. You will see that whenever there is a pretty defined segment, like it's like the quality score, this is the cutoff, but you see those segments are way over. See, the segments are over. The tops of the segments are the quality scores for this specific read in the nucleotide. Then what happens is something happened in the experiment, and you see that these segments went below 20. So here the quality became really poor, right? And remember, there's 10 billions of these experiments going on at any given time. That the total number of samples that were processed were 56, but the total number of samples, that some potential reads, nucleotide sequences, were several billion, all at the same time. Okay, and then you can see that as this went on, after something happened between nucleotide number 70 and nucleotide number 84, and then you can see that these segments, so that the top of it is the score, that became better. Something happened, artifact of the experiment, a machine broke down, the sensor didn't work, and this is just for one read, right? Now imagine 15 billion of these. Okay, and that gives you the idea of what that is. Now, this is a qualitative kind of a graphical look at this. Now, so what you will see here is that the color G, when a G gets added, is, or the G is that part of that sequence, which means a C got added, is black, A is green, T is red, and this, I mean, in real life, I don't know what those colors are, and C is purple. Now you'll see that each peak corresponds to each color. And you'll see that there's good confidence here, because these peaks are well defined. There's a little bit of overlap, and means that it could be that, it could be that, but still the sensor is picking it up in a very good way. What happens after 70 is that it's recognizing it as G and G. And again, sorry about this. It's you can see this G and G, and you see that peaks are there, but then there is one big overriding peak, right? A big broad peak is here, big broad peak is there, and so these are well defined peaks identified by a lot of G's, right? Um, uh, Guanines. You will see that the quality is low because you see some other guys overlapping, which means the system is not really sure which one really got added, though the peaks are shown. And then what happens is these competing peaks, they kind of go away here, and then you again see the sharp peaks without any errors, right? And so now you'll see that here the Fred quality score decreases below 20, which is this light blue line, and then here then it goes back up again. Again, we are looking at billions. If you remember that other place, N N N N N N N N means basically an unknown nucleotide. It was supposed to pick up something for that particular nucleotide that came from the sampling process, but nothing got picked up. Now let's look at the Fred quality score Q. So the Fred quality score is negative 10 log to the base 10 of that probability. Okay, now what is that probability? The probability is what is called a base call. A base call is the nucleotide which was added was that the correct nucleotide that was supposed to be added in that place. So if you have 90, now remember 90% is really bad. If you have 90%, which means you have a 10% error, which means 1 in 10, 1 out of every 10 nucleotide was likely an error. Okay, so then what happens? 1 in 10 is 0.1, right? 1 in 10, 1 divided by 10 is 0.1. So that's 0.1, which is 10 to the negative 1, right? So log to the base 10 of 10 is 1. Log to the base 10 of negative 1 is negative. Now go back to your simple rules of logarithms. So this is negative 1. So it's negative 1 times log to the base 10 of 10, and log to the base 10 of 10 is 1. So negative 10 times negative 1 is 10, which gives us this Fred quality score. Now that minimum requirement, okay, and this is now logarithmic, which means that blue line says that the minimum requirement is that you are allowed, and even this is not very good, you are allowed only one error in 100 nucleotides. One error in 100 nucleotides. And if you get, that means one error in 100 nucleotides means 1 out of divided by 100 is 0.01, and so 0.01 would be the probability of getting a right thing, right? So 0.01 is 10 to the negative 2, so that's negative 2 times log to the base 10 of 10, which is 10. So negative 10 times negative 2 is 20. That's how you get this number. That's how you get that blue line, which is an acceptable quality, which is a barely acceptable quality, the minimum. So then between 70 and 84, this has fallen below the standard. And before it's fallen the standard, that's kind of the problem with this. That's the problem with this particular quality. Okay, and so and so you will see that this gives you an idea of how these qualities are determined. Okay, any questions here? So now you know. Now typically what happens is then you kind of average over everything. Now a typical quality score for our experiment that we do on, it's called the NovaSeq, the instrumentation that we typically use, our workhorse instrument, and we've got some new ones, but that's the one that's been going on for the last 6 years, 6, 7 years. That NovaSeq instrument has, typically you will get somewhere between, like, your Fred quality score will be like between 35 to 37. Okay, 35 to 37 means that that's between 1000 and 10,000, so you will get like one error in 5,000. Okay, so that is considered actually is considered very good. So this was a very good illustration of what kind of what the notion of a quality score is. Any questions, any difficulties, any challenges, any thoughts about this? Okay. Now, I suspect that we won't finish today. We'll kind of go into a continuation, but I think this is it's nice to go into a it's nice to go into kind of next generation sequencing, having some of this information at your fingertips. Okay, so I think somebody might be knocking on the door. Somebody on the door?
Are you busy?
I'm in the class. What happened?
Oh,
No, no, no. What happened?
oh no, I just had a question
About
about because I looked at the diagnostic because I wanted to check my answers, and I just have one question about. I think one of the questions I got wrong was yours, and I
Can,
just wanna.
can you email it to me? When is the diagnostic?
Sorry?
When is the diagnostic?
Oh no no no! Like I looked at my answers
Okay.
because stupidly
Yeah.
allowed me, and I got one wrong,
Okay.
but I don't know. I just wanna make sure. I don't know.
Can you email it to me if you don't mind? I'm
Yeah.
in the middle of class right
Oh
now.
yeah yeah. Or
Okay.
when does your class end?
Class ends at another 15 minutes, 20 minutes.
Okay. Well,
Okay?
I'll just knock again.
Okay.
Okay.
You mean the class I gave you?
I'm still in the middle of class, Radhika. Yeah, sorry about that. Okay, so we'll see what is data analysis, and there are many kind of complicated data analysis. The software generally takes care of most of it, so you don't have to think too much about it. So, in this particular analysis, so for example, you will see here's some information, and I just got it from it tells you that it goes from 1 131 to 497. So it's almost like, what does it say? Without going into the specifics of it, it says that that so for example, you have a read, right? It's 100 nucleotides long. Where does it align? In what part does it align in the gene? So if it, the starting position of the read to that ending position of that read, where does it necessarily align the gene? And the gene will have 25 million nucleotide long. So here you will see that the first one is 497R. The R name is 1. That's not important. The position is 497. It starts at 497. That means 497 on that particular gene. This is the MapQ value. I'm not quite sure what that is, but it just tells you that how long is your how long is your read length. How long is your read length? So here you can actually see the read length is here, and then this tells you these symbols are the symbols associated with a these symbols are the symbols associated with the quality of each of these reads. So 37. Now this is the part that I was looking for. It's called the cigar score. Okay, and the full form of cigar score is compact idiosyncratic gapped alignment report. So, okay, it has nothing to do with the cigar, but I'll put that in the text, and you will see it's a compact idiosyncratic gapped alignment report. Basically, what it's trying to tell you is how does your uh, how does your read map against the reference genome, whatever part of the genome you have, right? And that's what, in this case, it's 37M, which means all 37 of the nucleotides, all 37 of the nucleotides in the read match perfectly against the nucleotides in the genes. And then this is, not sure what this is, but this is the position in the gene. Uh, I don't know what this is. Again, this is the sequence. This is the quality, right? The quality score in terms of the symbols. And then there are the additional tags which tells you more about that particular information. So let's go and look at a more close example of so let's say here is your read, ACTG, whatever. And let's say this, for example, is your position in the reference genome, gene, whatever, and it goes from 1 to 3, 4 to 19, right? So now what happens is you're taking your thing and in a pairwise alignment, just you did this, just when we did classical bio, just like when we did classical bioinformatics, your gene in your software is taking the gene, running around and doing all these matches. And remember, that's why you can also have errors in identification of the gene, and that is the that it's the long read sequencing are designed to ameliorate, to kind of mitigate some of these problems. Okay, so now what happens is, now remember, it's just like your typical pairwise alignment. It just happens several billion times in the processing of next generation, your next generation data. So this is your read. When you align it, you'll see that there is a gap here, right? There is a gap in the sense that from a reads perspective, you had an insertion. Reads perspective, you have an insertion, and in the reads, from the reads perspective, you have a gap here, which you put as a gap here, right? Whereas from the gene perspective, from the reads perspective, this is an insertion, this is a gap. Whereas from the genes perspective, this is a gap and this is an insertion. But it looks like for the most part it's a pretty good match. Now, the fact that it is not a good match could be because, you know, the particular information that you have does not meet the genome, because the genome was not necessarily a thorough representation of the human condition, right? So there are many reasons for that. So now what happens is the position. So now you look at the cigar score. Remember, in this case, the cigar score was 37. It means that all 37 of these nucleotides in this read mapped perfectly, without any gaps, without any insertions, without any deletions, none of that, right? So in this case, what happens is, so you'll see what the cigar score says. It says 3 matches. That means, and it looking at it from, obviously, from the read standpoint. So the first 3 are matching. So 3M, 3 matches, 1 insertion, because now you have an insertion because there's a gap here in the read, 1 insertion. Then you have 3 matches, 1 deletion, because that's really a gap, 1 deletion, and 1, 2, 3, 4, 5 matches. So once again, 3 matches, 1 I for insertion, 3 matches, another 3 guys, this GAA matches, then you have 1 deletion. Now there are some other things in addition to just matches, insertion, and deletion. There are other things that that are a little more complicated, and so those are also part of it. Again, if you want to know, you'll be able to find out a lot more about it. So once again, you'll see what these are, right? So in the analysis, the cigar score says match means alignment. BAM means it's a binary. So a lot of this information, the data is so huge that it's stored in a binary format. So it has a binary designation too. But an insertion means insertion, deletion, and then there are skipped regions and soft clipping, hard clipping, padding, sequence match. All of these are other designations that you could use, and we just looked at the more simpler ones. Okay, let's stop here. We'll finish next week, and then we will. I'll give you a homework, but the homework will be based on things that we have learned, and then we will do one more. Once we finish this, we have about 12, and then we'll just start on another topic, and then just move on from there. Okay. So, are there any questions? No questions? Okay, I'll send a homework. Somebody send me a reminder. I'll give you plenty of time to do it. So from that standpoint, is okay. And I'll try and grade everything by tomorrow. Okay. Michaela, you raised your hand. Did you have a question?
No, I'm just giving a thumbs up.
Okay, thank you. All right. I will see you guys next week. All right. All right. Take care. Bye.
Thank you.